Hello everyone. In today's video we're going to be talking about hemoglobin and transport of oxygen around the body. So just take a look at this diagram here. This is a rather lovely image of the hemoglobin molecule. So we can see here that the hemoglobin molecule is made up of 4 polypeptide chains. So hemoglobin has a quaternary structure. So it's a quaternary globular protein. So we've got these 4 subunits that are in these quite roughly tetrahedral shapes to form the overall hemoglobin structure. So in most vertebrates, it's an assembly of these 4 globular protein subunits, and each subunit is composed of a protein chain tightly associated with a non-protein prosthetic heme group. So that's this bit here. This is our non-protein prosthetic heme group. So the protein parts are the globin parts, and then the non-protein parts are the heme parts. So the heme group consists of an iron ion held within the ring, known as a porphyrin ring. So the porphyrin ring, which is in the middle, consists essentially of some molecules with the iron in the middle. So the iron ion is the site of oxygen binding. So each of these iron ions can bind with one oxygen molecule. And when it binds to one oxygen molecule, it temporarily goes from being Fe2 plus to Fe3 plus. So because there are 4 of these, one hemoglobin molecule can bind to 4 oxygen molecules or 8 oxygen atoms. Within each of your red blood cells, there are about 250 million molecules of hemoglobin. Therefore, that means that each of your red blood cells can transport about 1 billion O2 molecules, which is a pretty crazy number. So just a couple more key terms. So when hemoglobin is bound to oxygen, we call it oxyhemoglobin. And hemoglobin can either be saturated or unsaturated. So we say it's saturated when all of its binding sites are full of oxygen, and we say it's unsaturated when its binding sites have broken away from oxygen. So let's talk about oxygen transport around the body and how hemoglobin becomes saturated and how it gives up the oxygen at the tissues and organs at the respiring cells of your body. So this graph is called an oxygen disassociation curve. And it is showing you how saturated, so how full of oxygen, different hemoglobin molecules are. So percentage saturation of hemoglobin with oxygen at different partial pressures of oxygen. And notice that partial pressure is measured in kilopascals. But what does that term partial pressure mean? Sort of for our purposes, we can almost think of it as concentration, but essentially in any mixture of gases, each constituent gas, so each of the gases that make up that mixture of gases, has a partial pressure, which is the hypothetical pressure at which that constituent gas would be if it alone occupied the entire volume of the original mixture at the same temperature. So it's really just the pressure exerted by an individual gas in a mixture. So the partial pressure of oxygen really just means the amount of oxygen gas dissolved in the blood in any given area at any given time, right? So on this graph here, we've got these different kilopascals, these different partial pressures of oxygen. And we can see that hemoglobin behaves differently when it is in different partial pressures of oxygen. This is because of the way that it binds. So we're going to talk a bit about what this graph shows us, and then we're going to talk about what that means in terms of the binding. So at very low partial pressures of oxygen, the hemoglobin has given up all of its oxygens. Now, on a graph like this, it can sometimes be quite confusing, because if we think to ourselves, there are 4 potential spaces for hemoglobin to bind to oxygen. So it can be 0% saturated, 25%, 50%, 75%, or 100% saturated. They're the only options for a single hemoglobin molecule. And then if you look at this graph, you're like, well, how do we have like this one here, which is like basically 99% saturated? Because we're not looking at a single hemoglobin molecule. We're looking at like a whole certain volume of hemoglobin molecules. We're looking at a mass of hemoglobin molecules. So this is like a whole lot of hemoglobin molecules all together. So that's how we've got the curve looking how it is. We could also have a look at just one single hemoglobin molecule. So let's maybe just have a look at this one for a second, because it's a bit simpler, because it shows us exactly what we just talked about. So this one is if we're literally just imagining a single molecule, it's the same curve, but it's split it up into 0, 25%. It hasn't put a line at 50, but it could. 75% and 100%. So at very low partial pressures of oxygen, the hemoglobin is completely unsaturated. There are no oxygen molecules attached to any of the heme groups. Then, as the partial pressure increases, we can see an oxygen molecule binds on. And then as the partial pressure increases again, we can see another oxygen molecule binds on, another increase in partial pressure, another oxygen molecule binds on, and then, we need a really big increase in partial pressure for that last oxygen molecule to bind on. So there are a couple of things we notice straight away. Each heme group does not behave in exactly the same way. If they did, it would be a straight line on the graph. But it's not a straight line. It's a curve, showing you that in order to bind on the first oxygen molecule, you need quite a large increase in partial pressure of oxygen. But to bind on the second and third oxygen molecules, you need only small increases in partial pressure of oxygen. But then to bind the fourth oxygen molecule, you need a very large increase in partial pressure of oxygen. So let's just have a think about where all these partial pressures can be found. When we talk about areas of the body with low partial pressures of oxygen, we are talking about areas of the body that are heavily respiring. Every time oxygen enters that area, it's getting used up in respiration. So these will have very low concentrations of oxygen. I suppose if you lived a very lazy life, none of your tissues would ever enter this stage. But this could be when you're doing very heavy exercise, sprinting, going for a run, something where you're using up all the oxygen in your muscles. So we're really talking about your muscles down here. Where in your body would you find very high partial pressures of oxygen? You'd find this in your lungs. Okay, so at your lungs there is a very high partial pressure of oxygen. Maybe in tissues in your body that aren't really respiring very much, they have a really low metabolic rate, we're here. And then this part of the graph here is heavily respiring tissues. So as we move away from the lungs around the body, we start to drop off the oxygen. So here we are at the lungs. Okay, up here we are fully saturated with oxygen. So we're like, okay, I've got picked up all my oxygen, fantastic. And then I'm going to travel around the body. And as I travel around the body, I find that I go into areas of lower and lower partial pressures, depending on the organs and the tissues. Say I go to some skin that doesn't have a very high metabolic rate, maybe that's this part of the graph. But then when I go to some muscle cells, maybe cardiac muscle cells, they're really heavily respiring, or I go to some nerve cells, they're heavily respiring, and all different parts of the body will be somewhere on this graph. And so when I get there, I would give up some of my oxygen. So these different positions on the x-axis are representing areas of the body that have different metabolic rates and therefore have different partial pressures of oxygen. So why does the graph look like this? Well, let's imagine that we've got a hemoglobin molecule, and each hemoglobin molecule is bound to an oxygen, okay? I've all grabbed hold of my oxygens. So I'm fully saturated, I'm 100% saturated, I'm basically up here on the graph, okay? So I'm at the lungs. So then I'm going to start moving around the body, and as I move around in the body, I end up in areas with lower and lower partial pressures. But the binding sites for the oxygen molecules, when all four oxygen molecules are bound on, the binding sites face inwards, meaning that it doesn't want to let go. So in order for the oxygen to disassociate from the hemoglobin, you need a large drop in partial pressure. So when you end up here in an area of the body that has a low partial pressure of oxygen, now we've got enough of a pressure gradient for the first oxygen molecule to dissociate. So the first oxygen molecule leaves. The disassociation of the first oxygen molecule causes a change in shape of the hemoglobin molecules, shifting the binding sites. So now the binding sites are more facing outwards. They are now a bit less stable. They would more easily unload. They would more easily disassociate. So what we find is it only takes a very small decrease in partial pressure for the next oxygen molecule to detach. When that happens, it causes the same thing again. It causes a shift in the quaternary structure, exposing the third binding site, meaning that that third oxygen molecule, again, is much easier for it to disassociate. And so again, you only need a small drop in partial pressure of oxygen for this next oxygen molecule to dissociate and unload at respiring tissues. The unloading of the third oxygen molecule causes a shift in the tertiary structure of the hemoglobin molecule, basically hiding this third binding site again, pointing it inwards, making it much harder for that fourth oxygen molecule to detach. In order to lose the fourth oxygen molecule, you would need a large decrease in partial pressure for the fourth oxygen molecule to dissociate and unbind from the oxygen. This is really important. If it was easy to lose all 4 oxygen molecules, you'd leave the lungs and you'd go to the first organ and you'd just be like, there you go, have all my oxygen. So the nearest parts of your body to your lungs would be great. They would be able to have access to as much oxygen as they wanted. But then as you got further and further away down to the other parts of your body, you would have run out of oxygen. That would be no good. You wouldn't be able to maintain the metabolic rate of your feet or your hands or your liver. It's no good. So you need to have a way of going, you don't need this oxygen. I'm going to keep hold of this. Your partial pressure isn't low enough. I'm going to wait until I get somewhere that has a low enough partial pressure, and then I will unload my oxygen. So this curve, this sigmoidal curve, is important because it enables all of the respiring tissues in your body to have access to oxygen. And it is achieved by the shifting binding sites on the hemoglobin. So every time an oxygen loads or unloads, it causes a change in shape. So I described it as a disassociation curve. So I started off, they were full, and then they lost one, and they lost one, and they lost one. But it works exactly the same in the other direction. So you'd need a large increase in partial pressure of oxygen for your first oxygen to bind on, and then a little increase for your second, a little increase for your third, and then a large increase for the fourth. But it doesn't actually make that much sense to describe it that way, because at the lungs, where you're going to pick up oxygen, you've got loads. You've got a really high partial pressure of oxygen. So we tend to think of it as a disassociation curve. As we're going around the body, we're giving up the oxygens to our cells. This process is called cooperative binding. So we know that each hemoglobin molecule can bind up to 4 oxygen molecules, and hemoglobin exhibits cooperative binding. As oxygen binds, it increases the affinity of the hemoglobin for more molecule. And this increased affinity is caused by a conformational change or a structural change in the hemoglobin molecule. So let's just think about that. The binding of the first oxygen molecule increases the likelihood that the second one will bind, which in turn increases the likelihood that the third one will bind. So this is called cooperative binding. So the binding of one increases the affinity of the hemoglobin molecule for the next one and the next one. Now, one thing that some people can find a little bit confusing about this graph is that we imagine that during the travel around the body, we end up at 0. You basically never end up at 0. it would be very odd for your body to be in this area. That last oxygen atom very rarely disassociates from your hemoglobin. It's like an emergency oxygen that you would give up if you found yourself in a situation where there was suddenly a very, very low partial pressure of oxygen, perhaps in your muscle tissue, but that would not be a common occurrence. So, if you're sat here reading this, it's not like your hemoglobin is going around giving up every single one of its oxygen. It usually keeps hold of the last one because it never reaches an environment with a low enough partial pressure to give it up. So we've talked then about the oxygen dissociation curve, so what it looks like, we can describe it, and we can also explain why it looks that way, and we can also explain why that matters, like why that is beneficial. Let's take a look at this graph. So this graph is showing us what looks like the same thing. So we've got partial pressure of oxygen in kilopascals on the x-axis, and we've got the percentage saturation of hemoglobin with oxygen on the y-axis. But then we've got three curves, and these three curves are showing us how hemoglobin behaves differently in different carbon dioxide concentrations. So if we were to sort of take our control, like, okay, medium, normal circumstances, medium carbon dioxide concentration, this one in the middle, we can tell ourselves, okay, this one is what we're like on average when we're not really heavily exercising, but we're also not asleep. So in what circumstances would we find ourselves in a situation with a high carbon dioxide concentration? Well, you know this. This is in a situation where your respiratory rate has increased. So your cells are undergoing more respiration and they're producing more carbon dioxide as a waste product. So a high carbon dioxide concentration or a high partial pressure of carbon dioxide in an environment is an indicator that you are doing lots and lots of respiration. So maybe we are looking at muscle cells while you are running. Whereas a low carbon dioxide concentration is an indicator that you have a really low respiratory rate. So maybe we are looking at tissues that are not doing very much respiration, they just generally have a low respiratory rate, or maybe you are sleeping and none of your cells are doing that much stuff. So we can see that in these circumstances, the curves are shifted. In high carbon dioxide concentration, the curve shifts to the right, and in low carbon dioxide concentration, the curve shifts to the left. But what does this actually mean? Well, it means that at a particular partial pressure of oxygen, so let's take an example, let's say like at 8 kilopascals of oxygen, if there was that particular partial pressure in your environment, each of these hemoglobin molecules would have a different saturation level. So, the hemoglobin molecule that found itself in the high carbon dioxide concentration, let's say that's about 45% saturated. The hemoglobin in the medium environment, let's say that's about 80% saturated. And the hemoglobin in the low carbon dioxide concentration is 90% saturated. So in the same environment, so in the same partial pressure of oxygen, they have different levels of saturation. And that makes total sense because when a hemoglobin finds itself in a high carbon dioxide concentration, that is an indicator that there is a high respiratory rate here. Lots of carbon dioxide has been produced and therefore lots of oxygen is needed. And so the hemoglobin disassociates or unloads more oxygen to provide those cells with oxygen for its increased respiratory rate. This is an adaptation, whereas in normal circumstances, we are like, okay, medium carbon dioxide, medium respiratory rate, I'm going to keep hold of most of my oxygen, whereas low carbon dioxide concentration is an indicator that the cells really don't need any oxygen. They have a low respiratory rate. So shifts to the right mean that the hemoglobin is disassociating more readily, and shifts to the left mean that the hemoglobin is disassociating less readily. So the next question we have to ask ourselves is why? Like on a molecular level, why? Well, we know that hemoglobin is made of protein, and we also know that carbon dioxide dissolves as carbonic acid. So carbon dioxide will dissolve as carbonic acid and it will lower the pH of the blood. So when the pH of the blood is lowered, this will affect the structure of the hemoglobin. Let's just think about what we know about protein structure. We know that small changes in pH can destabilize the molecule, and that's exactly what happens here. When it becomes more acidic, the hemoglobin is less able to bind onto the oxygen, so it more readily gives it up. So decreasing the pH decreases hemoglobin's affinity for oxygen. So decreasing pH, i.e. increasing the carbon dioxide concentration of the blood, decreases hemoglobin's affinity for oxygen. Affinity basically means how readily or how strongly it binds to the oxygen, meaning it will unload oxygen more readily, so it will become less saturated more readily at a particular partial pressure of O2. If you increase the pH, i.e. you have less dissolved carbon dioxide in the blood, this will increase hemoglobin's affinity for oxygen. Therefore, it will be more saturated at a particular partial pressure of O2. This is known as the Bohr effect or the Bohr shift. So then, thinking just about protein structure, we can readily answer this question. How do you think temperature would affect the affinity for oxygen? Well, we know that increasing the temperature destabilizes proteins, whereas decreasing the temperature increases the stability of proteins. So at higher temperatures, the hemoglobin is going to bind less readily. It's going to have a lower affinity for oxygen, and it is going to more readily give up its oxygen at tissues. So it would cause an increase in temperature would cause the curve to shift to the right, and a decrease in temperature would cause the curve to shift to the left. This also makes sense, because in rapidly metabolizing tissues, a lot of heat is produced as a waste product. So higher temperatures are an indicator of a higher metabolic rate, just like higher carbon dioxide concentration and lower pH are indicators of a higher metabolic rate. And remember, we've referred to carbon dioxide as an indicator, and then we talked about carbon dioxide is linked to pH. But also, another metabolic waste product that is an indicator of a high respiratory rate is lactic acid. Lactic acid is produced when cells are forced to go into anaerobic respiration, and we also know that lactic acid would decrease the pH further decreasing the affinity of hemoglobin for oxygen, increasing the rate at which hemoglobin readily unloads its oxygen at the tissues. So this is a really clever system. Hemoglobin sort of doles out the oxygen, have a bit of oxygen to all the cells. But then when it comes across a cell and it's like, oh, this cell's clearly doing a lot of respiration, I'm going to give it loads of oxygen so it's not forced to go into anaerobic respiration. So it's not forced out of aerobic respiration. So that's exactly what this graph is showing you here. So if we imagined, just for the sake of argument of this one, that pH 7.4 was normal, average, medium levels of pH, and then as the pH drops to 7.2, and remember this can be because of carbonic acid, it can be because of lactic acid, the curve shifts to the right. And an increase in pH, so less carbonic acid, less lactic acid, the curve shifts to the left. And again, that means at a particular partial pressure of oxygen, we will see different levels of saturation. So at pH 7.2, we've got, whatever this one is, whereas at pH 7.4, we're much higher saturated. And then at pH 7.6, we're even higher saturated again. So there's quite a lot of language that we've used here. We've talked about loading and unloading of oxygen. We've talked about percentage saturation of the hemoglobin molecule. And we've also referred to hemoglobin's affinity for oxygen, i.e. how tightly it binds onto that oxygen in particular conditions. So hopefully all that makes sense. So just again, this is a disassociation curve. of oxyhemoglobin as the pH changes. And this graph is just showing you what we were talking about earlier, about how changes in temperature will affect hemoglobin's affinity for oxygen. So we can see at higher temperatures the curve is shifted to the right, and at lower temperatures the curve is shifted to the left. So we can see that at higher temperatures, hemoglobin's affinity for oxygen drops. So increased blood temperature reduces hemoglobin's affinity for oxygen. That means that it unloads oxygen more readily at the tissues. So we mentioned the Bohr effect earlier, but let's just formalize that. So when we have more carbon dioxide and water are dissociated into carbonic acid, which then further dissociates into hydrogen ions and hydrogen carbonate ions. So increased partial pressure of carbon dioxide is directly related to a lowered pH. When the pH rises, hemoglobin loses the H+ ions from the amino acids in its structure, causing a subtle change in conformation that makes it bind more readily to oxygen. But when the pH falls, hemoglobin picks up H+ ions that were created during this reaction with the carbonic anhydrase, and its affinity for oxygen decreases. So that's what causes the Bohr effect, causes this shift in the stability of the molecule, a shift in its conformation, its shape. Okay, so in the next part of this video, we're going to be talking about different types of hemoglobin. You might want to take 5 minutes rest just to clear your head from all this listening to all this information. I've given you loads of information so far. And not just that, you might want to spend some time just going over this stuff, checking you understand it, testing yourself, and then coming back to learn about myoglobin and fetal hemoglobin. Okay, so hopefully you took a little break and your head is all clear and you're ready and you're excited to learn about the next part of oxygen disassociation and the transport of oxygen around the body. We're going to be learning about 2 types of hemoglobin. The first is myoglobin and the second is fetal hemoglobin. But let's start with myoglobin. So what is myoglobin? Well, myoglobin is very similar to hemoglobin. It is an oxygen binding protein. In comparison to hemoglobin, myoglobin is a tertiary protein as opposed to a quaternary protein. What that means is that it only has one heme group. So it only has one heme group, one globulin group, as opposed to hemoglobin's 4 of each. So myoglobin binds to only one oxygen molecule, whereas hemoglobin binds to 4. So myoglobin is a protein that's found in the cytoplasm of skeletal muscle tissue in vertebrates in general, and it's found in almost all mammals. So myoglobin is pretty distantly related to hemoglobin. Compared to hemoglobin, myoglobin has a much higher affinity for oxygen than hemoglobin. And it doesn't have that cooperative binding with oxygen like hemoglobin does, because there's only one of them. Now, the function of myoglobin is quite different to the function of hemoglobin. The function of hemoglobin is to transport oxygen around your body, but the function of myoglobin is to store oxygen. Interestingly, myoglobin was the very first protein to have its 3-dimensional structure revealed by X-ray crystallography. And this was in 1958. And the guy that discovered it, a guy called Kendrew, got the 1962 Nobel Prize in Chemistry for it. So it's an extremely well-studied protein in biology, but we don't know as much about it as you would expect. So myoglobin has a much higher affinity for oxygen than hemoglobin. And the job of myoglobin is to store oxygen until the muscle cell is exercising heavily and is desperate for some oxygen. It's to tide the cell over in that really short space of time where it's no longer receiving enough oxygen from hemoglobin, and the cell doesn't want to have to go into anaerobic respiration. So only in those times will it give up its oxygen. So it only gives up its oxygen at very, very low partial pressure. So it's got a really high affinity, and the curve looks quite different for myoglobin to how it looks for hemoglobin. So let's take a look at the graph to compare the two of them. So we can see here that they look different. So we have the same volume or mass of myoglobin and hemoglobin, whereas hemoglobin forms this sigmoidal curve, myoglobin forms a hyperbolic curve. So it gives up, it unloads its oxygen at much lower partial pressures. So we can see the partial pressure at which it becomes 50% saturated is much lower in myoglobin than it is in hemoglobin. At the same partial pressure, myoglobin would be much more saturated than hemoglobin. And this is due to its role in storage of oxygen as opposed to transport of oxygen. So diving mammals such as whales and dolphins have really high concentrations of myoglobin in their muscle tissue. And it's this high concentration of myoglobin that enables them to be able to dive because they can store so much more oxygen. Myoglobin is 10 times more concentrated in the muscles of diving mammals in comparison to human muscles. So that's really interesting. So we find it quite hard to pack proteins together inside our cells because when you have loads of proteins together, they can start to clump. This can be massively problematic. It can cause disease. But what researchers have discovered is that in diving mammals, there has been evolution, there have been mutations and changes in their myoglobin to make the amino acids on the outside positively charged. This means that they repel each other. So all the myoglobin molecules, they've all got this positive charge. They repel each other, so they're pushing each other away. So having all these positively charged myoglobin molecules stops the proteins from sticking to each other. And this is how it enables diving mammals to have loads and loads of myoglobin in their cells, whereas our myoglobin doesn't have a positively charged outside. So we have a limit to the amount of myoglobin that we can have in our muscle cells because otherwise it would all clump together and it would be massively problematic. What's really interesting, I think, is the scientists who've studied this have shown that this trait of having positively charged myoglobin has evolved separately. Like this isn't something that all mammals had at the beginning when they became diving mammals, because lots of diving mammals evolved separately. And this is an example of convergent evolution, when different organisms solve the same problem in the same way. So they independently evolved this myoglobin with more positively charged amino acids on the outside, which I find really amazing. And it's such a clever fix for that problem. You obviously don't need to know about it in that much detail. I just thought that was an interesting aside. So let's finish this off with the last type of oxygen carrying molecule, and we're going to be talking about fetal hemoglobin. And you know, it's obvious from the name that fetal hemoglobin is found in fetuses. So fetal hemoglobin starts being produced at around 6 weeks of pregnancy, and then the levels of fetal hemoglobin remain high after birth until the baby is about 2 or 4 months old. So fetal hemoglobin is also found in babies, and then slowly, as they grow, they will start to replace their fetal hemoglobin with adult hemoglobin. So fetal hemoglobin has a different composition from the adult forms of hemoglobin, and it has a much higher affinity for oxygen than adult hemoglobin. This is really important because the mother's blood is going to travel past, so the capillaries containing the mother's blood are going to travel past the capillaries containing the fetal blood in the placenta. And if they had equal affinity for oxygen, the oxygen would not travel from the mother's blood into the fetus's blood. The fetus has to be stronger. The fetal hemoglobin has to have a greater affinity so that it can take the oxygen from the maternal blood. Imagine if the fetal hemoglobin had a lower affinity for oxygen than the maternal hemoglobin. It would never get any oxygen. So it has a much higher affinity for oxygen. So you need to think to yourself, okay, is my sigmoidal curve going to shift to the left or shift to the right? So as you see, fetal hemoglobin curve shifts to the left. Fetal hemoglobin has a higher affinity for oxygen compared to adult hemoglobin, and therefore at the same partial pressure of oxygen, fetal hemoglobin is more saturated. This means that at the placenta, oxygen will move from the adult hemoglobin to the fetal hemoglobin and give the fetus the oxygen it needs in order to do respiration, to release ATP, to use for growth and development. Okay, so that was a lot. Well done you. Well done for concentrating. Lots to think about, lots to learn, lots of new key terms, lots of new key processes, and lots of phrases that I've talked you through. So take your time, draw it out, make sure you understand the graphs, making sure you can describe what each graph would look like and why it looks like that. Make sure you can make comparisons, make sure you can describe the Bohr effect. Why is the Bohr effect happening? You need to think about, well, what's causing this change in pH? Think about the role of carbon dioxide and lactic acid and carbonic anhydrase and the effect that that has on the hemoglobin. Well done for now, and I will speak to you very soon.